Yeah, so my name's Kevin Hall. As I say, I'm an Illumina employee. Actually worked for Illumina for about 18 years now, mostly in product development, but more recently working with collaborators on workflows and scaling workflows. So we had a fairly major event for Illumina last week, which some of you may have seen and heard about, but I thought I'd recap some of that because I think it's very relevant to sort of the topic of the conversation here of how we do newborn screening at scale. So we talked last week, and Francis D'Souza, our CEO, talked about this new platform called NovaSeq DX. And this has been about 5 years in the making. It's our next sort of high throughput flagship product. And it's taken many person years of research to bring all this together. The way the Illumina works, and probably a lot of other companies, we have a lot of like technology groups. And each of those groups have been working hard on their particular technologies, and as they become mature and are ready, we bring them all together and integrate them into to new platforms. So just to highlight some of the things that we have worked on, as most people know, we use flow cells, and these are pattern flow cells, and we've been able to bring these nanowells much closer together. That's what the patterning is much tighter, so that we can vastly increase the amount of density, the number of clusters that we have. To do this, we've also had to improve what we call our base calling algorithm. So we've got to pull that data out of those nanowells, to record what's in there in terms of the type, the base. We've speeded up things a lot. So there's new sensors, new stages involved, so we can actually scan. Unfortunately, I didn't bring any props or anything, but we have 8 lanes, and we can scan each of these lanes, top and bottom. It takes about a second with sort of nanometer precision, and that's it's pretty tolerant, to keep everything in focus. I think one of the major advances for the whole platform as well is, and it's something that we heard a lot about, was that the amount of packaging that's associated with sequencing is quite high if you're shipping with dry ice packs and things. So the whole supply chain, we need to improve our green credentials essentially. And so we've done a lot of work on reformulating the chemistry such that we can actually freeze dry or lyophilize some of the key components. And this means that we can actually ambiently ship some of them through a fairly wide range of temperatures. And then once you get them sent to you, you can put them in the fridge or the freezer as appropriate. Other advances are with, in terms of analysis, we put the DRAGEN cards onto the instrument. We did this with the previous product with NovaSeq 2000, but obviously there's a lot more data involved with this. And all the advances that have happened with the DRAGEN analysis systems are built into the platform. Just a few of the sort of the highlights here of the configurations that we've got. We're launching early next year the NovaSeq X Plus, which is actually takes 2 flow cells along with that middle flow cell there, which is 3 terabases per flow cell. The instrument on the right actually does 2 flow cells, so that you get 6 terabases per run. And then later in the year, we'll launch the 1 flow cell configuration. That's the one on the left, with the other 2 flow cells. And I suppose for high throughput, you look at the one on the right there, which produces 8 terabases per flow cell, 16 terabases per run, which equates to about 128 genomes in 2 days at a price point of around $200. Just some of the metrics that are involved here, that as you increase the going from comparing it to the NovaSeq, the current platform, the NovaSeq 6000, the output's gone up 2.5 times roughly. And similarly with the number of reads, if you're doing counting applications. The throughput, sort of genomes per hour, has gone up 2.5 times. So previously you got 1 genome per hour. With all the advances that we've got, we get 2.5 genomes now per hour. As I said, we've moved from 48 genomes per run to 128. I'll emphasize a little bit about the packaging, because I think this is relevant. If we're thinking about scaling, it's not just the workflow, it's just all the other things that are involved. So what we've done is reduce the number of cartons that are needed, so that's like a real the 3 boxes on the right is what's needed for doing NovaSeq runs. The box on the left is the new NovaSeq X, what's needed. and we've reconfigured everything such that the plastics that are used are different. You can recycle, you can deconstruct things, you can put the right plastics in the right bucket. There's recyclable plastics. We're using biopolymers more. The buffer cartridge is made out of a cane sugar polymer, I believe. And as I said, with the ambient shipping, we don't use any dry ice anymore, and you don't have ice packs to get rid of. And I think that's important, both as individuals, with our green, reducing our green footprint, but also for institutions where they have targets for sustainability and things. A little bit about the library side. So there's that little insert there, the picture there that shows what's put on the instrument. You load up your libraries. Each of the lanes are separate, so you can do separate experiments in each of those lanes. It's the configuration or the structure is such that you can also put that insert onto an automation platform. So if you're doing things at high throughput, that's all configurable, and you can just transfer that onto the instrument. A little bit about the data quality. Data quality is sort of key. It's what we live and die by at Illumina. We need to make sure that we maintain our quality standards for the data coming off the instrument. There's just a few examples there. I mean, we've worked a lot on things called phasing and pre-phasing. That's the way the enzymes work, and how the fidelity, how quickly they go on and off, and how well they do that. And that's all enabled us to bring error rates down, in fact, and reduce incorporation times, which means that we can speed the runs up and get more data off them. So I mentioned the DRAGEN system. So that's built on board the high throughput instruments. It's got all the latest sort of DRAGEN advances that have been developed separately with DRAGEN Graph and the machine learning. At launch, there'll be certain applications supported around whole genomes, exomes, and transcriptomes, but more will follow as we work things through. Some of the other things that will really help with the data is the ability to reduce the data footprint as well. So built in is this Aura data compression software, which is a company we purchased a few years ago. And this enables us to reduce the FASTQ file size down by 80%. The whole DRAGEN side, the analysis on board is flexible in the sense that you don't need to use it. You can stream straight to the cloud, or you can do pretty much everything on DRAGEN, or some hybrid, whatever works for the particular configuration. There's also other sorts of software associated with the product, and I think one of the things, I suppose, hopefully that'll come out as we talk more. It's no good just having a box. You need to have boxes that fit into workflows. You need to develop workflows that work end to end. That's that term, or sample to answer, in a seamless way. And so you need to have the right hooks and work through the upfront side of things in terms of library prep and management of samples through things like Clarity Limbs, or the Limbs software, all the way through to the back end, where you've got analysis on board or in the cloud, and then how do you handle that data at the back end? You're going to store all this data. You want to do things with that data at the back end. It could be queries, federating data, and so we're trying to put this whole package together and make it flexible, but at the same time it works. Everything's connected. So just to wrap up, so with the new NovaSeq X Series, it's been sort of worked on to improve efficiency and productivity, and obviously improve the amount of genomes that you can get out of systems as people want to scale more. You can use that scaling to do deeper studies or broader studies, and then there's the sustainability effort as well to reduce that side of things. So I'll stop there.
Well, thanks, Kevin, for kicking it off, and thanks to the organizers for inviting Nanopore to the event. I think we're at like a great inflection point in genomics in general. Like, I'm sure Stephen and Robert and many others in this room thought about us already being here many years ago, but it's taken some time to get here, and the field has really changed. We're at this true inflection point where lots of sequencing has happened. Illumina has made and paved that way. It was great to be part of that at one stage. But now the tides are changing. It's a better understanding, what are we going to do with all of the data? What information is it providing? How do we get it closer to the clinic? How do we make it accessible to more and more people? And I think that's where we'll start to see the investments moving into the field moving forward. And it truly is a really exciting time to be involved in this. And so as I go through this, we've had about 20 years since the first human genome was published, 15 years of Sanger sequencing, 15 years of short read SBS sequencing. And if one thing comes out of this is that there's more work to be done. We don't truly understand all the variation that's out there today. The telomere to telomere consortia certainly highlighted that and pointed out that from their perspective, there's 8% of the entire genome that was just not sequenced before. And that's truly been a limitation because of the technology in interrogating those complex regions of the genome. So now we're at a point where we can start to sequence those regions of the genome that have not been interrogated before. But the critical point is to better understand, what do those mean in a clinical context? And I think as we move forward, we sequence more, that's going to become ever more apparent. And then that will feed into programs like newborn screening. So it is truly an exciting point. And of course, Nanopore being part of that telomere to telomere consortia, enabling the mapping of those complex regions. And it's not just the missing 8% of the genome that wasn't discovered before, but it's actually looking at the complexity of the genome that was there. You know, there was indications of regions that were challenging to interrogate, like CNV structural variants. That's all now possible because we've made such advancements in new technologies. And so for those of you that don't really know about Nanopore, I'll try to put it into layman's terms. We like to use this analogy of kind of seeing the genome in full color. So if you look at the Beatles picture, we are an Oxford-based company, so hence the correlation to the Beatles. But wouldn't you rather be looking at the genome in full color, or not a copy, as you see here, the CD versus looking at Abbey Road in real time? If you think about Nanopore technology, it is truly looking at native sequencing capabilities, a single molecule sensing technology. So you read the actual DNA and RNA strands. There's not a copy of that. The advantages of doing that, because you not only read that DNA and RNA, you get to start looking at base modifications and other things that come along with the DNA. So everything that makes us up is not just DNA variations. There's a whole lot of other environment around that plays into the mechanism of diseases. And with Nanopore, you can start to interrogate that. Now I know that we have traditionally been kind of pigeonholed into a long read platform. That has changed. We are an all fragment platform. So you can do short read, long read, take your pick. It's really flexible. Operationally, it's seamless. And so there's a lot of accessibility to the platform that we can now enable. And it's quite portable too. There's not a need for a large instrument. If we start to think about how we get genomics into point of care, there's a seamless transition from those large instruments to the smaller ones, all with the same technology, leveraging Nanopore. And so as the video shows here, that is a strand of DNA going through our pore, hence the name Nanopore. And then what we call the squiggle coming out is just the raw read signal that we translate into the standard ACGCT signal. But along that, we have algorithms that can interrogate the modifications like 5-methyl-C, 5-hydroxyl-C. And I'll go through some of the reasons why that's going to become important and why we should think about that context in newborn screening. So traditionally, we have kind of sequenced to death SNPs, indels, and we do that really well with the current technologies out there today. But there's quite a lot of, as I said, missing. So if you're looking at methylation in real time, syndromes like Fragile X, where we know there's an indication of methylation playing there, those are completely missed. Structural variation, yes, you can see some structural variation in current technologies, but it's challenging and difficult to read. And copy number is another one. There is growing amount of evidence of the diseases that are related to that, like Duchenne's, for example. If we could truly understand the implications of copy number variants, which I think we're only at a tipping point of really understanding what's out there today, It requires that we have new technologies to interrogate that. And certainly at Nanopore, we believe we can provide all of these variant types with one assay. Just looking at the variant characterization overall in an entire genome. So this depicts a publication from Evan Eichler that showed that the percent of variant bases across the genome of what's currently interrogated today is actually a small proportion. If you just look at the SNPs and indels, that's only about 15% of the total genome. So you're missing all of this other information, let alone layering on that methylation piece. Put that all together, you're looking at about 84% of total variation across the genome that's just not being realized today. And just thinking about how that will imply to disease, we don't know yet. There's a lot of research that needs to take place to better understand and characterize that, but that's where we need to go. So you don't have to believe me. There's a lot of publications out there that highlight these. So the Mayo Clinic showed and confirmed that 8% genome missing. CHOPS took that even further and looked at the genome in the bottle and said, actually, there's about 19% of the entire genome that just is not read completely. Decode showed that 40% of structural variants are missing. And most studies that are out there today leveraging methylation are just using an EPIC array that is only looking at 3% of the total epigenome. There's 97 other percent out there. And what that really highlights is, and the reason why a lot of the diagnostic rates out there today are still relatively low. There is most, as it says here, most Mendelian diseases, 50% are undiagnosed. Ever more so, there's reasons to start looking at the advantages of long-read technologies. So this just highlights really quickly one of the areas that may be of interest when you think about newborn screening, looking at SMN1 as a particular gene of interest. Nanopore technology can uncamouflage that as part of the dark genome and read through that very well. The other one, CR1, which is an Alzheimer's disease, may not be so relevant in newborn screening, but it just shows that with the long-read technology, you can start to interrogate these really challenging genes of interest. Now, there was a lot of hype, and I won't belabor the point about the record-breaking Stanford approach, particularly with Stephen in the room. But to me, that study is not the important part. It's not about the speed. It's about the fact that they actually increased the diagnostic yield by 12% with comparable SNP accuracy to other technologies today. The important thing here is that with this one patient, they waited 2 weeks for their standard of care and got variants of unknown significance. Within 8 hours, they actually identified the variant with Nanopore technology and were able to change the care management that this patient already went through. So that's the real value, is understanding the more complex regions of the genome that you can actually put into standard practice. To take that one step further, Danny Miller in Seattle, he had a patient, their sibling had a known genetic variant, disease. And so they wanted to know at birth, did his sibling also have that? So within 3 hours, they were able to sequence the sibling and actually identify that they didn't carry the variant, they weren't a carrier, and that family could sit at ease. That all happened within 3 hours. And that's largely because they knew what they were looking for, but they had the technology that allowed them to do a full human whole genome in less than 3 hours of sequencing. So we're at a point where we can really translate the improvements of not just long-read technology, but real-time sequencing and accelerate the advancements of genomics into the clinic as soon as possible. So I'll just leave you with the fact that Nanopore sequencing is fast, easy. From an operational standpoint, it's multifactorial in that there's no batching, it's one for one, it's really accessible, it is quite portable, so please come to our booth, you'll see a small version of it. And it's information rich, which I believe moving forward when we start to think about the implications of disease and newborn screening, we need that full breadth of the genome to understand. So thank you.
Yeah, thank you, Kevin. Thanks for joining us. What we wanted to do in the next sort of 25 or so minutes was have a bit of a discussion around the different sequencing technologies that we've heard about and that are out there in the context of this conference. So thinking about this in the context of newborn sequencing. What I'm going to do is start by doing some introductions so that we all know who is with us. And then we're going to have a conversation here amongst the panel, but also if people in the audience have questions they want to ask, and also people online, do let us know, and we'll come to you as well. So Kevin and Tonya, we've heard from you. Meredith, do you want to just introduce yourself?
Yeah, of course. I'm Meredith Ashby, and I am the Senior Director of Strategic Marketing at Element Biosciences.
Thank you. And Jeff?
Yeah, hi, I'm Jeff Henno, Segment Associate Director in Segment Marketing at PacBio.
And Stephen?
I'm Stephen Kingsmore, and I head up the Beginnings program, which is one of the newborn sequencing efforts.
Great.
And I'm at Rady Children's Hospital.
Thank you. And I'm Richard Scott. I'm the Chief Medical Officer at Genomics England. You'll hear from ourselves and from Stephen and others about the programs we're all doing. We're at the moment in a design phase of a newborn genomes program as well. So we've been sort of thinking lots, and lots of the people in the audience have been thinking about what the opportunity here is, and what the world that is in front of us thanks to all of these advances in technology. We've heard a bit from Kevin and Tonya already about some of the particular ways of thinking that have been going on at Illumina and Nanopore. Meredith and Jeff, and then in a moment I'll come to Stephen. It'd be really interesting to kick off by sort of thinking about how you're approaching this, what the particular challenges you see ahead, where the sort of the angles you feel that we should be sort of particularly chipping away at.
Yeah, well, I'm glad I get to go before Dr. Kingsmore and not after. I would say that from our perspective, the number one challenge with newborn screening is increasing the number of brand new people who we can help get a strong start to life. And that means increasing access, but also increasing the number of genes that we can screen for, including things that are more rare but very serious. And so from our perspective, that really comes down to controlling costs and decentralizing, right? Low costs lets us screen for more genes, and decentralization devolves control over testing to local communities and local health centers so that they can be sure that they are prioritizing the needs of their specific regional populations. So Element wants to be a part of that by bringing low-cost sequencing and a benchtop platform. So it's basically benchtop model, right-size batching, but at factory scale pricing. But really, we're here to listen. You all are the absolute experts in this, and I hope to talk to many of you over the course of the next couple of days and learn what's most important to you.
Great, thank you. Jeff?
Yeah, so from our perspective, I can tell you what our customers are telling us in the screening world. And they basically, with current technologies, conventional methods, they see recurring issues and recurring difficulties with particular genes that we all know. And so they come to us to see, okay, what can a long read approach bring us here? And we've seen oftentimes that they increase their detection capabilities. Those tough genes are better covered, either with a targeted approach. This can be amplicon or capture, or with the whole genome approach, obviously. What we also hear from projects that are running big numbers of whole genome sequence is that, yes, as was already pointed out before, that there is a section missing in that genome. And so it seems to our customers that long reads will be able to cover those gaps. We call them dark regions. They have other names, but from our perspective, long reads will be able to cover that. And then the question is, okay, how deep do you cover? How deep do you sequence? For that, we have seen a 30X that is excellent. It's the Mercedes-Benz of the sequencing. The T2T has shown that very clearly. But then as you go down in coverage, of course, cost becomes more interesting, and the throughput. So we're looking at solutions also, rather than having a targeted approach, do it whole genome. You may have heard the announcements at AGBT, where also a packed biogenome is a complement to the big volumes that Illumina is running, basically.
Thank you. And so, Stephen, you'd be interested in your perspective. And I guess I know one of the things that we all who are sort of planning these programs are thinking about, the absolute sort of critical steps in this, that the most major bits that we need to unpick. I'd be really interested in your perspective.
Thank you, Richard. Wow, this is an amazing time. 2022 is going to be one of those famous years in history. We have the convergence of three things. So we've heard about next generation sequencing and how the $100 genome is finally within visible sight. That's amazing and opens up immense opportunity. And one of those opportunities is the fact that 140 million babies every year are screened for inherited condition. In some countries, it's one condition. In some, it's as many as 80. So what an opportunity to transform healthcare delivery. But there's two other areas of technology that are also blossoming, and one is IT, to make possible actually organizing the information, parsing the information on the same scale. And the third is a transformation of drug development for orphan disease. Those three are coming together to make possible a new world of hope for children with genetic diseases.
Fantastic, thank you. So I think Tonya and Kevin would be interested in sort of any further reflections that you have while I'm giving you a bit of time to have a think. In terms of the audience, I think we've got some roaming mics that are ready to come to people if you have questions. So do raise your hands. There's also the distinct danger that we will just chat away, which leave us to if you want. But put your hands up early if you want to have your chance. So why don't we just, Kevin and Tonya, if you've got anything further you just wanted to add, and then we'll go to question number one in the audience.
Yeah, just a couple of things. Obviously, my mic on. Okay, yeah. In the time allowed, I couldn't cover everything. So there is an awful lot still going on with Illumina in some of the other areas that I didn't get to cover around. With DRAGEN, especially, we're looking at sort of targeted callers, so the ones which are difficult to find. We can look at those and do big improvements and see what we can get out with short reads. We've also got some long read technology coming out. I think it's the beginning of next year, which should help a lot more to sort of fill in some of the gaps. And then on the benchtop side, I think we've talked about putting this new chemistry. It used to be called Chemistry X, it's called XLEAP now. And we're going to be putting that on our NextSeq 2000 platform 2024 before that comes out. But there'll be some, I think it's a 500G 3 genome product for sort of that sort of mid-range if people need that.
Thank you. Tonya, anything extra to add, or we'll come to you?
No, I think we've kind of covered it from a technology perspective. For me, it's more about the ethics around doing newborn screening and the implication of knowing your genome from birth, and how do we manage that? How do we manage data over time? And what do we disclose at that period? It's certainly with long reads, a lot of information, and it becomes how much information should we be sharing?
No, and I think we'll hear lots today on sort of various more technology aspects, and then tomorrow I know we'll hear from all of the different programs who have been thinking about lots of those questions as well. As you say, it's this convergence of sort of multiple leaps in technology together with then the ability and the importance of engaging and working out how we best use those and in the right way to dig into that. So let's go to the audience, and I think when we have questions, it'd be great to hear who you are, where you're from, and your question.
Hi, I'm Crystal. I'm a geneticist working at the American Civil Liberties Union, and I think that everything I've heard has been incredible. I finished graduate school in 2020, and it seems like even in just these 2 years, there's been these huge leaps forward in technology. But kind of jumping off what Tonya was just saying, at ACLU, our big concern is genetic privacy. So I'd be really interested in, how you're, and I guess the different companies' privacy stances and how to protect this data, and kind of that overall belief in how to monitor the security of this data over time, considering how, I guess identifying it is inherently.
Great, thank you. Kevin, should we start with you?
Yeah, just a quick, absolutely. We have to, it's paramount that we keep privacy, front and foremost. So we need to create these sort of trusted research environments and things like that. So with the databases that we work with and the way we construct all of our software, that has to be in there. There's a lot to be done, and discussions with the thought leaders in the area to make sure it's done in the right way, that you can give access, but at the same time you protect patient privacy. But definitely yes.
I guess a sort of perspective from me, and like anyone else in the panel who wants to jump in, and I might also sort of get maybe Stephen to share some thoughts on this. I think the question of both sort of data privacy, but also balancing that with the benefits of making data useful to make sure that we address these really key questions. So that, for example, when we're thinking about using genomic technologies to in a screening context, that we're moving forward and we're making sure that all communities can benefit from the benefits of screening, and we're not just skewed towards communities who have been traditionally more represented in genomics programs. And that real balance of that pivot point of sort of privacy and bringing those benefits and finding the right way of engaging in those questions, which are both sort of deeply sort of human and often quite personal, both to people, communities, and sometimes quite different between different countries. And then thinking about the deep geek tech of what's even possible. And I think finding that sweet spot is what I think is one of the most exciting things about these programs, but also the most challenging. You'll hear, I'm sure, from us and other programs about some of the ways we're thinking about that, but doing that in conversation with both sort of sequencing organizations, but also those sort of thinking about how data is stored and so on is crucial. And I know, Stephen, you guys have been thinking a lot about that and how one supports that. I don't know if you've got a perspective.
Yeah, just had a couple of comments. Thank you. Great question. Great starter question. It's a big issue, and it's one we need to keep very near the top of the list and not forget it. We actually all met, all of the groups who will be presenting over the next couple of days, for a full day yesterday, funded by the Liberty Fund, to have a dialogue. And this was one of the core issues that we spent a good chunk of the day working through and comparing notes and addressing what the solutions were. I do think one really important thing to point out is that we have digital electronic health records. And they contain ridiculously sensitive information about our personal history. And a genome is going to be part of the electronic health record. So it's a new digital information set, but it's an extension of the electronic health record. And I think everything that we've done to maintain privacy of electronic health records will tie then to the genome. And that's a good model for us to think about. If we're okay with, let's say, our psychiatric history or our infectious disease history being stored in an electronic health record, that's probably a good starting point to think about how we ought to manage genome information as well.
Great. And so I don't see any other hands right now. So I'm, and also can't see any online yet, but do put your hands up and wave if you have questions. I guess sort of linking to that question about data and sort of balancing both privacy and sort of the comparability of data so that we can really make the most in terms of the potential of this technology. One of the things that that is, both is challenging, particularly in a sort of competitive and diverse sequencing environment, is thinking about that comparability and thinking about linking things up. So it's both sort of from, before the machine, from the machine, and then through pipelines to sort of interpretation. And I'd be interested from the panel, so which elements of that are they most sort of focused on as companies, or where do you think there's sort of almost sort of missing activity in the broader market to make sure that we don't have sort of data in silos by technology, for example? Yeah.
I could say, well, I think you bring up the point of silos is the important thing to think about. We have talked for years about federated data stores and how we're going to enable that. I think there's a few companies doing that, but I think there needs to be more. And programs like National Newborn Screening, it's not just about the clinical implication, but how can you use that for research and then share that? And we, let's face it, need to start sequencing beyond just your average white male. We need to start going into more diverse communities and making it accessible. Therefore, think about how we share that. At Nanopore, we're really focused on one, generating that complete genome perspective, but making it accessible to those more diverse communities. And how is that interpreted? So we are working and looking for partnerships on improving the interpretation of those results. Part of it is that we know the value is there in the genome, but it's extracting that data from the genome. And that's really where our focus is. That will be not only from our own development of taking that raw signal to something of value, but then how's that interpreted and shared more widely?
Yeah. So, from my perspective, different countries are in such different places with regard to this. I think Public Health England is such a standout. There's very few places that have that kind of universal access, and also a coordinated government system that allows for the consolidation of different data types and really allows for population-wide learnings, which can be shared with the rest of the community. I think, and I think we all saw the benefits of that during the COVID-19 pandemic. I think in the US, the public health system made some big strides in the area of sharing data and consolidating data. And so perhaps we can use that success story as a way to further it and expand it to different application spaces, including newborn screening. But I also want to point out that not everyone has access to the fleet of high throughput systems that are available at Rady Children's Hospital and at Public Health England. And as everyone has noted, it's very important to start to get more screening accessible in countries outside of just Western Europe. And I think a big part of that comes back to increasing access by lowering costs and decentralization, right? So, and again, I think some of the mid-throughput platforms that are available today, including ours, could have a role to play here in doing, maybe not the whole exome, maybe not the whole genome, but doing a right-size panel that's right for the community at sufficient scale that it is in fact affordable. And so that other folks outside of these massive, the places where they're most advanced are able to start joining the revolution here and benefiting from it.
I think there's two strands. I think there's a dimension of interoperability and having a smooth workflow so that you can generate the data in some sort of standard way. So that, I think that's down to providers and working out what that looks like. And then there's the second part, which is you have all this data and then you've got to want to federate it and bring in other data from other techniques. But they're trying to drive the standard and bring all that seems it's a challenge, but probably needs addressing.
So just to flag to people, we've got 10 minutes left. I think we've got a question up there on the side. So get going with your questions if you want to squeeze them in.
Hi, everyone. I hope you can hear me with the mask on, if that's okay. So my name is Ankit. I work for Amazon Web Services. I'm the genomics lead for the public sector. And we are very proud of the work that we're doing with Genomics England, Illumina, and several of the other folks on the panel there. I wanted to go back to the question, Dr. Kingsmore, that you raised about integration with electronic health records, and wanted to see how, maybe pick your brain about future forward-looking thinking, because the challenge with EHR systems, not the implementation, but generally speaking, is that you create silos even within a healthcare platform. right? For example, the pediatric department is not talking to the cardiology department. Within the same hospital, there are glass walls. What kind of like future systems or future vision that you have, maybe to the panel, about truly democratizing access to this kind of data for research uses?
Wow, okay. I'm now regretting I'm sitting on this panel. Great question. I think this is a big problem. I think you're right that electronic medical record systems, while we love them, we also fervently hate them, and we know their limitations, and we need to solve for them. And I think that the rare disease space with 10,000 diseases, and the fact that each of these diseases is rather rare, is a unique space that really, right now, modern medicine doesn't do a good job with. And these poor families go through hell with their children, with their affected members. And we need a new solution. And it does need to bring together data. I am a big fan personally of seeing, can we build a system around the cell phone that's parent controlled, and then ultimately will be controlled by the child when they reach age that they can make decisions. And can we have this be a family directed resource to allow those families then to manage a lot of the information and actually help their doctor find the disparate information. So that's my vision. How to get there will require a lot of thought, and hopefully AWS will be part of the solution.
Thanks, Stephen. Interested in whether any of the other people on the panel.
Yeah, to add to that, it's an interesting concept, right? The portability of the genome. It's our core set of instructions, so why not have it on us? And I think Apple is spending a lot of budget and time on this aspect, right? So I think that's the future indeed, I would agree. Why not have it on you and then run apps to analyze whatever polygenic or rare variants that you want to look at?
So one of the questions I have in terms of sort of how this plays out, we're in the early part of the journey here.
I noticed you didn't take that question on, Sneaky.
I think I would have just echoed what you said, Stephen. So one of the questions I have, and we had this discussion yesterday to an extent, which was about there are different ways in which genomics can play a role in screening in newborns. One can think of it as sort of something which is absolutely integral to the current screening, sort of sitting alongside, complementing two assays, maybe multiple assays, all playing in together and triangulating. You can also imagine a future where it works slightly differently. I'm interested, given that we're sort of got a particular focus on technology here, from the panel, how you guys are thinking about that sort of multimodal, if you like, approach of sort of bringing in other data types, so that it's not the genome alone, it's other data types that can help triangulate and give that sensitivity or specificity or both.
I think that's fundamental in Nanopore, and I think I highlighted that in the talk. We don't think just about the genome, we think about RNA, we think about methylation, and bringing that all together in one approach. The future, I think, of Nanopore 2 and other companies, frankly, what do we do around proteins and that interaction between transcriptions and what results in the protein? So how do we bring all that data together? That's going to be the future.
Yeah, I think that's a great. I wonder if we're all going to say the same thing here. Yes. The underlying technology of the Element platform, it's basically a detection a system for any biomolecule. And we have scientists in R&D now who are very excited about adapting it to detect other kinds of molecules, be they protein or metabolites or what have you. Obviously, it's a DNA sequencing platform today, but this is something that we're actively exploring.
I think it's the same with Illumina. I think, yeah, it's we have multi-omics groups as well that are looking at these different areas.
Same with us, RNA-seq, the next frontier. Yeah.
Great, thank you. And I think we've got a question, we've got two questions maybe. Let's try and fit both of them in. And then the next one is, I've seen at least, is just here.
Hi, Apni Santani. I'm a Chief Medical Officer at Veritas Genetics. Thanks for a great talk, and I'm enjoying the questions as well. So now that the technologies have evolved to the point where the price of the genome is very accessible, one of my question is really related to content. We know that there are these founding principles for any population screening program. And when we start leveraging any of these powerful technologies for newborn screening, some of the challenges are related to not knowing enough about their prevalence or not having a confirmatory test when a pathogenic variant is released. And I'm sure, Richard, you and Stephen have thought a lot about this. I'm just curious to know what evidence-backed strategies are going to be used to determine what diseases get screened for a newborn screening program.
Yeah, so I think that's a fair one for perhaps myself and Stephen to start with. And I know that different people, and you'll hear more about it in the talks from individual programs. I think a lot of this is that this is not a new challenge. The concept of screening in newborns and screening more broadly has been around for a long time. So actually building on these fundamental principles, the Wilson and Jungner criteria are the foundation. I know of the work that we're doing at Genomics England and the work that Stephen and others is doing, and then thinking what that means in the context of genomics or potentially sort of multi-omic approaches. So there are some sort of tweaks to it, but at the bottom of it, it comes down to sort of that balance of sort of thinking about, beyond the cost, but to the acceptability and so forth. And really, I think all of the programs that we've been talking to are taking a very similar approach, conservative in the same way that sort of, newborn screening programs more broadly over the years have taken. And that feels like the right approach. This is, there's an opportunity brought by technological advances, but it's not a new sort of opportunity in the sort of broader scheme. I don't know, Stephen or others, if you
just
want to
maybe add the comments. I totally agree. So Wilson and Jungner, 1968, World Health Organization, those principles have stood the test of time. And I think every group that's participating in this space is buying into that as those are the guiding principles. I would say, though, that for many, many of the diseases that may have an effective therapy, we don't know quite enough about the natural history of the condition, such as the incidence that you mentioned, to really know yet whether to put it onto a panel. And so this will be adaptive, I believe, where we will do our very best to identify conditions, knowing full well that some of the initial conditions may not really stay on the panel as we accrue data, and we will remove those and others will join. So we want to start with a strong foundation, adhering to principles, but also keep an open mind and future-proof. And let's remember that there are now hundreds of new molecules of various types and platforms in development, which will be hugely beneficial for individual diseases that today are not tractable.
Yeah. And likewise, to today's theme, responding to the changes in that technology brings in terms of what we can detect, and we know what is possible today, but sort of knowing that's going to continue to evolve. Now, I think we've actually reached time for this section, so I want to thank the panel for joining me up here to discuss this, to kick off the conference. I've really enjoyed it, and thank you to the audience for some great questions.